Working memory in stroke patients. This study examines the effect of stroke on working memory performance. It is the result of conflicting information recorded in a previous study on working memory deficits in amnesia patients. Quantitative research involves the use of numbers to test a hypothesis using hard data. This data can help predict outcome by using measured amounts and determine validity through values. This is a strong research methodology which reveals immediate results without subjecting those results to researcher bias. The effects of stroke on working memory performance. So our independent variable is stroke, and the dependent variable is memory performance, spatial, object, and binding. The study participants completed a computerized delayed match to sample task for working memory in which 3 objects on a grid are presented by different locations. All locations could be used by the 5x5 grid except for the center block. The participants are exposed for 1,000 milliseconds or 1 second, followed by a 3-second delay. They are then asked to identify a match or the location of the object. In the case of the object condition, the symbol is placed in the center of the grid, and the participant then must decide if it was a match to the object presented in the trial. In the spatial condition, the object is a specifically located black dot, and in the binding condition, it is an object at the specific location. The schematic overview of the experiment is after the exposed and delayed period, a probe is presented to the individuals. In this slide, matching probes are shown for the 3 different conditions: spatial, object, and binding. For the binding condition, a non-matching probe is a recombination of the object and a location presented in that trial. In the subsequent memory task, long-term memory for objects and bindings is assessed. All participants scored above chance level, or what is expected if the results were random. Results indicate the sample of stroke patients in the chronic stage do not reveal working memory deficits on a standard neuropsychological test, but they clearly do show deficits on an experimental working memory task. An important discovery is the working memory impairment concerns a single feature conditions, not the associative condition, which is the stroke. The results imply associative working memory may be moderated by structures of the medial temporal lobe, which concerns long-term memory and is not affected during the stroke on either side of the brain. The quantitative research method used in this study is its strongest attribute. The weaknesses pertain to the small number in the cohort and the single instance of study. A larger group of participants combined with longitudinal study would increase the reliability of these findings. Changing these 2 variables would not completely alter the study's simplistic and straightforward design. Incorporating fMRI results and other anatomical information would provide much more depth but impact the study.